as we mentioned earlier, cytokinesis is the division of the cytoplasm and the final step in the cellular replication. Cytokinesis is equal to, or it is the division of the cytoplasm. So it's going to divide the mother cell's cytoplasm equally between the two daughter nucleuses. This cytoplasm must be encased in a plasma membrane. This is going to enclose each daughter cell in its own individual plasma membrane. A lot of times this phase, cytokinesis, is going to begin early on. We can actually see the first couple hints of it early on in anaphase. Now, the interesting thing is about cytokinesis is that it's going to proceed differently in plant versus animal cells. It just depends on the shape and the structure of the overall cell as a whole. What is one of the key characteristics, or think about it before you move on to the next slide, what is one of the key characteristics between plants and animal cells, specifically the outermost layer, like the cell, the outermost boundary between the cell and the outside environment? All right, in animal cells, cytokinesis is going to go as follows. It's going to start off by a cleavage furrow that's going to appear between the two daughter nucleuses. That's going to be sort of this little indentation right here. This is going to be ultimately formed by a contractile ring of actin filaments. Remember, actin filaments are going to be those small little fibers that are found or play such an important part in our cytoskeleton. They're going to form a tight little dense ring right around the middle of the cell, and they're going to start to squeeze and pull until it actually pinches the mother cell in two. That is the little cleavage furrow. It's kind of like putting a drawstring around I don't know, a slice of cheese. And you just slowly pull that string tighter and tighter and tighter until it pinches off and separates that block of cheese. This is how we can do it with animal cells. Now, plant cells are a little bit different. Plant cells don't have just a soft cell membrane. They do have a cellular membrane, but they have some other feature, or one other feature, that prevents you from actually just pinching off the cell membrane, and that would be the cell wall. So in the case of a plant cell, we have a rigid cell wall around the outside, so we're going to have to separate our cell or undergo cytokinesis slightly differently. In this case, we're going to do it with the formation of a cell plate. A cell plate is just going to be a series of small vesicles made by the Golgi apparatus that are going to line up right down the center of the cell. They're going to fuse into one thin vesicle extending all the way across the mother cell. Inside that vesicle, we're going to start to just build up a brand new cell wall. The space between those daughter cells becomes filled with middle lamella. The daughter cells will later secrete primary cell walls on opposite sides of the middle lamella, which will cement the primary cell walls together. So here's just a little bit of what that looks like. We're going to start off by fusing these vesicles are going to be created by the Golgi apparatus. They're going to fuse together and they're going to form one great big long plate right down the center of the cell, and that's going to join up with the cell membrane just inside of the cell wall. Once we have separated the two halves of the cell, we're going to then build up a cell wall in between them. So that is cytokinesis in plant cells. Please let me know if you have any questions or comments.